Hello, sir. You can start now.
All right. Very good morning, everybody. Today we are going to have this live session about mass spectrometry-based quantitative proteomics. So as you are aware that for most of our biological problems, we want to compare some conditions, right? For example, how a disease condition is different than the healthy condition, or how a drug treatment can induce some proteins in response to that given drug, or because of a given stress condition, or any kind of thing which you want to test out. We always look at quantitative changes, right, at the proteome scale. Looking at 1, 2 protein and their quantification is much easier. But when you think about at the proteome scale, looking at thousands of protein, it's not so straightforward, right? So today we are going to give you this concept of quantitative proteomics using mass spectrometer and different ways of how to analyze that type of data. So what I thought I'll just kind of start with a little bit basic about proteomics and how to separate the complex proteome. Then I will walk you through about mass spectrometry-based proteomics workflow. Throughout, my focus will be more from the lab sessions, how we can show you the technologies and the mass spec configuration from our lab. So as some data analysis. And then finally, I'll talk about quantitative proteomics, few concepts, and again, some hands-on session. So it will be like more a mix of few key concepts, which I will try to cover from my slides here. And then our lab people, the TAs of this particular course is going to talk to you about some of the workflows and the live session. So just to refresh you, although of course all of you are already aware of that, moving beyond genomics, now leading toward the understanding the transcripts and the proteome and the metabolome, more functional entities, we have more and more level of complexities, right? So proteomics looks for, let's say we have 20,300 or so genes of human, which we are aware of from Human Genome Project. We have many isoforms, followed by we have a lot of post-translational modifications, and then we have these functional proteoforms, right? So the numbers for these could be a lot, could be to the a million, right? Which we are not even exactly sure about this number. So therefore, as you can understand that moving from the genome, uh, moving forward to the looking at the proteome and the post-translational modifications is quite complex. So therefore, studying proteome and proteomics is not very straightforward, and we need different workflows for analyzing that type of work. So proteomics, while it's almost, we have made good progress in the last 2 decades, but the draft human proteome maps were deciphered not so long ago. It's only a decade ago, when the first draft of the human proteome map was deciphered with the mass spectrometry-based approaches by 2 different groups from Bernard Kuster and Akhilesh Pandey team, which showed almost 70% of the proteome can be confirmed using the mass spectrometry. And then next year in 2014, antibody-based approach was shown by Matthias Uren's team. So for each protein, they generated antibody, and they showed that again 70% of the proteome map could be deciphered from the tissue. So again, these were kind of the initial leads. First time, like Human Genome Project, we got the draft human proteome map. But these were, just to remind you, only 70% or so proteome, and it's still the data analysis guidelines were quite, not so stringent that time. So therefore, the Human Proteome Organization, which looks for the big activities of the proteomics and the Human Proteome Project, a consortium looked at the draft human proteome map in 2020 during the COVID time. And again, this was now released as the map of 91% human proteome map, including our group from IT Bombay contributed in this activity. And now we have less number of missing protein, it means almost 9%, which is the numbers that keep getting reduced over the time. But still, we don't have 100% complete proteome for which we have the experimental evidence that these proteins are present in human. So as you can understand, it's quite challenging field to work with. And therefore, we need a variety of ways of looking at how to analyze this particular complex sample. So for proteomic study, one can look at different workflows. Everything starts from the protein extraction from the cell. And there are different protocol one need to try out, different lysis method you need to try out to ensure that every other thing, for example, the carbohydrates, lipids, nucleic acid, salt, they all have to remove out from the sample. And only thing which will remain will be the protein, and that has to be also removed with the salt. So this is the first key consideration for looking at the protein extraction at the proteome scale. Then you want to separate the protein, and traditionally people have used the two-dimensional gels, gel-based approaches, but also one could move directly without gels with the gel-free approaches with the mass spec, and again separate the complex proteome using different principles, which I'll talk more in detail. And then aim is to identify this protein, what we have in our complex proteome mixture in a tube, and then protein identification with good confidence for every protein. And if you were to compare your control with the disease condition or the treatment, then one could look at the quantitative proteomics change. And finally, once you have identified some good leads and the targets and the biomarkers, you can do further characterization of these targets. So this is kind of broadly a proteomic study involves. Typically, a workflow traditionally people have used after protein extraction, they use the 2D gels, followed by identifying the protein on the mass spec. But now there is more and more approaches when the gels are not being used. Directly, the samples can be analyzed with the mass spectrometers, and then followed by the bioinformatics, protein identification, and quantification. So these are the standard workflows of looking at the proteome. So while the focus for today's session is not on the gels, mainly on the mass spectrometry-based quantitative proteomics, but I thought it's still good to remind you that this is one of the oldest technique of proteomics, two-dimensional electrophoresis, which involve two principles looking at isoelectric point and the molecular weight. For this, we utilize the isoelectric focusing unit based on the pH of the protein, and when the pH becomes pi, the isoelectric point becomes 0 over there, net char 0, and therefore you can separate the proteins in the IEF field based on this principle. Followed by, you can use the SDS page to separate proteins with the molecular weight. And once you have done both the dimension separation, you see this kind of pattern, which shows you a lot of spots on the gel, and every spot represents the pi value and the molecular weight. This was done almost 50 years ago in 1975 by Cloth and Farrell. But a good start that, given that proteins are very complex, we need to involve different principles for separation. This was the one beginning of that. Later on, building upon from this kind of concept, in around 1998, a technique came which is known as the MUTPIT, multi-dimensional protein identification technology, which involved again two principles of chromatography, the strong cation exchange and reverse phase to separate the proteins without gels using chromatographic principles. So this background gives you the where we are in the proteomics to begin with, and then how proteomics then started moving toward the mass spectrometry-based workflow. So mass spectrometry-based workflow is now currently the major workhorse of all the proteomics work. And we have different type of sample, whatever you want to analyze in your given lab for your research work. You can take any sample type, extract the protein, digest the protein to make the peptides. Then you are going to ionize these peptides using the electrospray ionization, followed by the protein separation with the complex mass analyzers, followed by the data analysis, the chromatograms, and you can then get the protein list. So this is kind of the a generic workflow for the mass spectrometry-based proteomics workflow. In this process, there are different components of the mass spectrometer, which I said nut and bolts of mass spec, which are important for you to keep in mind. For example, your proteins are in the solution form, which have been digested to make peptides. These peptides are separated with some chromatographic principle. So sample inlet comes with the chromatography, followed by, now these are in the liquid phase. You want to ionize the peptides in the gaseous ionized form. So therefore, ionization techniques are required. Electrospray ionization or ESI and MALDI, which is matrix assisted laser desorption ionization. These are techniques to do the ionization part. Then next, you want to separate these ions in the mass analyzer using different physical principles involved. and the different type of mass analyzers are used for that. After that, then you are looking at detector to detect these ions, and then followed by the chromatograms which are being generated. You're going to subject them with some databases and search for the proteins and analyze the data. So this kind of the nut and bolts and the broad view of the mass spectrometry in general. The first part is important, which is the ionization source, where your peptides in the liquid form has to get ionized form and go inside the mass analyzer. So therefore, we involve various tricks here with the very high volt, with the pressure differential, with the charge differential, so that all the ions which are being formed, they should go move inside the mass analyzer. And scientists which gets credit for this thing, John Fenn, who got the Nobel Prize for electrospray ionization. And this particular cone which you see, the plume, that is known as the Taylor's cone from the scientist who found this phenomenon. And therefore, this entire thing is known as soft ionization, which means with the minimal fragmentation of the ions, one could move all these ions inside the mass analyzer. So therefore, this is the very first and important step. Second part is now how to separate these complex ions, these peptides, which in the solution form have moved to the gaseous ionized form. And now they are traveling in the mass analyzer. There are multiple options for the mass analyzers. We have quadruples, we have time of flight, we have linear traps, ion traps, we have orbit traps, ion cyclotron resonance. Many of these things are available for us. So while there is not a theory lecture here that I can not talk to you about all the details, but I thought to touch upon this, at least with some important key concept, why we are looking at so many mass analyzers. So ideally, we look at least 2 mass analyzers, because your ions which are moving from the electrospray ionization in the first mass analyzer, let's assume it is the quadruple, over there, you are looking at all the intense ions, and then you are selectively doing the collision of those ions, and then you are taking another mass analyzer when you are fragmenting each one of these ions. For example, the blue one gets fragmented, then the red one will come and get fragmented, likewise the green one will come and get fragmented. So this is known as the MSMS. If you had only one mass analyzer, that was the only MS1. If you have 2 mass analyzer, that is MS2. This is known as the tandem mass spectrometry. And therefore, you need to look at a configuration of the mass spec when you have at least 2 mass analyzers available. For example, QTOF. It is one of the very popular configuration of mass spectrometer, where you're utilizing 2 principles of quadrupole and time of flight. So in quadrupole, you have the metallic rods where the ions are going to move in the magnetic and electric field, followed by they will travel in the time of flight path, and they will get further separated, and therefore you have the QTOF, right? So I hope you are clear. We have the ESI, electrospray ionization, as ionization source, followed by the QTOF, 2 different mass analyzer, which is tandem mass spec, and therefore we got the ESI QTOF as one of the configuration of the mass spec. Likewise, then people realize that they need to do much higher resolution of the separation of these ions, and this new technology came, which is the orbit trap, which is really a leading technology from 2010 to 2020, when the ions are oscillating around the central electrode in the Z-axis, and it really provides very high mass accuracy and the high resolving power. So this is another configuration. Sometimes these configurations are even with the 3 mass analyzer, like in the orbit trap fusion, for example, we have quadruple, ion trap, and orbit trap. 3 mass analyzers are there. Triple quadruple is another popular configuration, where people utilize this for the targeted proteomics and targeted metabolomics based workflow. Again, idea is to have the one set of rods as the quadruple 1, another set of rod as the quadruple 3, and in between we have collision cell as the Q2, which is misnomer. So idea is you can select the various intense precursors, fragment those, and generate the MSMS spectrum for a specific peptides. So this is something which is more utilized for the validation of the target in the targeted proteomics. I hope this gives you, like, there are a variety of possibilities of mass spectrometry configuration. You are now at least theoretically a bit familiar with the mass spectrometry based workflow, the ionization source, the mass analyzers, possibilities and need for different mass analyzers because of the tandem mass spectrometry utilization. And now from the lab, we are going to show you some of the mass spec configuration, so you will be able to understand it much better. But all theory which I talked to you, now you can see practically from the lab. So over to the TAs now. We have 3 TAs. Surbhi, Amit, and Ankit. These are all very good PhD students from IT Bombay. They are the TAs of the course as well, and they will be showing you these technology live from the lab, and then I'll continue further from there. All right, Surbhi, over to you.
Yeah, thank you, sir. I hope I'm audible to everyone. So I'm Sylvie, and I'm going to show Orbitrap fusion to all of you. As Professor mentioned, that it is a tribrid mass spectrometer. It has an Orbitrap, quadrupole, and ion trap. So this is the nano LC, wherein we put the samples. So as you can see, this auto sampler comes out and we put the samples in these little vials and we put it in the auto sampler, and from there is an injection over here from which the samples are picked up. So the samples are picked up, and you can see over here which all steps are going to happen. There's sample pickup, loading of the sample, gradient preparation, and everything is visible here. You can see the flow here also. Since this is a nano LC, the flow is in nanoliters per minute. Here is the solvent of the nano LC. This is solvent A, which is 0.1% Fa, and this is solvent B, which is 80% ACN. A gradient of ACN and 0.1% Fa is set, which helps in elution of the peptide. So the nano LC is connected to the mass spectrometer using these nano wipers. This is a pre-column, which is also called the trap column, and then this is the analytical column. This is where the actual separation of the peptides happen before going into the mass spec. Now you can see inside of this is the ion source. Here the ionization of the peptides happen when the peptides elute out of this analytical column. If we zoom into the camera over here, is it spotlight? So we can see in this camera over here, this is the tip of the column, and this is the start of the mass spec. It is called the ITC. So the peptides are eluted from here, and from here they enter the mass spec. Now I'm going to show you how a chromatogram looks like. So once the data has been generated, this is how a chromatogram looks like for a complex protein sample.
Maybe I would say, like, please wait for a minute. Let them fix the issue. I think your screen is not seen probably. Now it is seen, so you can repeat your previous steps as well briefly.
Okay, so as I was mentioning, this is a tribrid mass spectrometer. It has a quadrupole, orbitrap, and an ion trap. Now, this is the nano LC, which is connected to the mass spectrometer. And I'll show you the auto sampler. Here we put in our samples in these small little vials. The samples go in. We put in here, and as you can see, there is an injection here from where the samples are picked up. So this, if you can see this pointy thing, it goes inside the vial and it picks up the sample from there. And this is the interface of the nano LC, where everything, all the processes, all the steps which are happening is mentioned. The flow rate is mentioned. Since this is a nano LC, the flow is in nanoliter per minute. You can also see the pressure in the tubings over here. This is the solvent. Here is solvent A, 0.1% Fa, and this is 80% ACN. A gradient of solvent A and B is set up, which helps in the elution of the peptides. And the nano LC is connected to the mass spec using these nano wipers. As you can see, the nano wipers are very thin to maintain the nano flow rate. Here first comes the pre-column, which is also called the trap column. It is to prevent any impurity or any blockage or contaminant going directly into the analytical column. And this is the analytical column where the actual separation of the peptides occur. Now, you can see the tip of the analytical column over here in the camera. And this is the start of the mass spec. So from here, the peptides are eluted, and here they get ionized, and then they enter the mass spec. Now I'll show you a chromatogram of a complex protein sample, and once the data is generated, how it looks. So this is how generally a chromatogram looks like. You can see that we have run a 120 minutes gradient, and the elution has started a little after 10 minutes. So you can see that there's a little, except for this intense peak, there's a little Gaussian pattern over here, and very dense peaks you can see. And if I zoom into these peaks, you'll see the resolution over here. So these are the individual peaks. And over here, you can see the MYZ ratio. And on the x-axis is the relative abundance of the peptides, and on the y-axis is the relative abundance of the peptides and of the ions, and on the x-axis is the time. So this is how a chromatogram looks like. And further into this lecture, we'll see how we can analyze this data. Now, next, Ankit will show the next instrument. Amit will show the next instrument.
Okay. Okay. So hi all. So as we have shown the orbital fluid which uses three different mass analyzers. Now I'm going to give you a short demonstration of Q exactly, which uses two different types of mass analyzer. Like in orbital fusion, it uses the quadruple, the orbital and the ion trap. In case of QE, it uses the quadruple and the orbital mass analyzer. So now this is the QE system. So in the orbital, it uses the LCMS. In case of QE, we are now using a HPLC system. So right now we are doing metabolomics and lipidomics here. So basically two different mass analyzers are the quadruple and the orbital. So basically quadruple is basically used as the mass filter. It's based on the M by Z ratio. It selects the top intense precursors, and those intense precursors will further went to the orbital mass analyzer for further fragmentation. So in QE, the resolution of the orbital mass analyzer is like almost 1,40,000. In our updated models is like something like 2,40,000. So now, if I show you the HPLC system, so this is the system here, and in this particular area, different columns. The columns is situated here. So in case of lipidomics samples, the CACH C18 column is used. In the different type of modes, we need to switch between the columns. So you can see the column here. Now coming to the solvent part, we for lipidomic samples we use 4 different kinds of solvent here. So here we use the 2 different modes, the positive mode and the negative mode. So in positive mode, we basically try to see what are the protonated ions are there. It could be protonated metabolites or it could be protonated lipids. And also we use a negative mode where we try to see the deprotonated ions. It could be metabolites or it could be of lipids too. So in positive mode, we use 2 different solvents, the solvent A and the solvent B. So in solvent A, there is a solvent A. We basically use 1.1% of it that we increase the protonated forms. But in solvent C and D, that is used for the negative ion mode, we cannot use any kind of effect. So right now, so here is the auto sampler here. So in auto sampler, we try to we basically put the samples in this kind of small vials. You can see in both of the studies, when metabolomics and lipidomics, we use these kind of inserts. We put the low amount of volume here, and we try to put this in the auto sampler, and we try to run so that run the samples. All right. Now, coming to the softwares. So there are different softwares we use for the analysis. One is the Excalibur, where you can see the total amount of total chromatogram here, where the x and y axis are the time and the radio abundance. In case of metabolomics, like we use a gradient of 20 minutes, and in case of lipidomics, we use a gradient of 32 minutes. So based on that, it tries to measure that what are the different lipids and metabolites are getting identified. Another of the software is the Excalibur, where we try to put the sequence of our query, and we try to give the positions from where the machine is going to take the samples. So so now over to Ankit, who is going to talk to about the OLTIS. So as Survi said, Survi was talking about orbital fusion. Then Ankit is going to give you the demonstration for the TSQ OLTIS.
Hello everyone. So good morning. So I'll now, after showing you the two mass spectrometers, which uses one are two mass analyzers, hybrid configuration like quadrupole and orbit trap. Now I'm going to show you one example of like which Sarah has already shown in the which Sarah has already shown, like the targeted validation is a triple quadruple mass spectrometer where it uses two quadrupole. So here the configuration is like quadrupole followed by a collision cell chamber and followed by another quadrupole. So now it is again coupled with the chromatography is the same HPLC or high performance liquid chromatography for the small molecules that was with the QV. So here again the same configuration that you want to be providing in the autograph the samples, and they will have the column for chromatographic separation of the analytes or the metabolites or the proteins or the peptides, followed by you will have the solvent system where you will give the hydrophobic gradient that will ensure that your peptides or molecules are segregated and run into the system. Now I'll just quickly shown into you the different parameters or the methods that you have got a brief glimpse while I was speaking. So here you can see that is the ion source, that is HESI, so electrospray ionization. I think you have seen. So here we need to provide the source for that. Then we have like the spray voltage in the positive ion and the negative ion. So the ionization requires the huge voltage. Then we have like the different gases that will help the from the liquid to ions when it is formed, to how the ions will move into the analyzing system, followed by the temp. So this is the ion transfer capillary tube that leads you to the insertion of the ions within the source or the mass analyzer. So after this giving, we need to give the scan parameters. So here I am showing you one of the examples. So in the targeted or the in this configuration. So in both the things, what is generally required is that we give the precursor ion within the defined M by Z. The precursor ions are selected either in DDA mode or in DIA mode. When the precursor ion is getting selected, then it is getting fragmented and is analyzed by the second mass analyzer. So for the peptides, both these fragments gives us the identification of the peptide, and for the metabolites also the MS. After the MS1 precursor ion is selected, the MS2 ion gives us the unique fragmentation spectra for this particular identification of metabolites. But when we are going for the targeted thing, so we need both the precursor and the product ion that we need to monitor. That is known as transition. So if I just can show you in the method. So in the method you can see that we are giving both the precursor and the product. So this is the two transitions. So in the first quadrupole, it will be just filtering out this particular precursor M by Z, and then it will filter out after in the passing through the collision cell. So which is we are giving the collision energy voltage. It will try to find out the particular M by Z. So these two transitions are monitored, will be monitored for a particular compound or a peptide. And the start and the end time is the chromatographic gradient that we are keeping. And here, if we try just now focus with you, so this is the quadruple 1 resolution and quadruple 3 resolution. That's how much delta we are allowing for those to pass away. And now after this, we give the analysis. And if I just show you the gradient, so this is how we give the gradient of 20 minutes if we are giving. So how the hydrophobicity is increasing. We are increasing the hypothesis. Now, after the run is done, we get this kind of analysis where you will be finding out that at what retention time. So in this x-axis, it is retention time, and y-axis is the intensity. So in this particular, at what time it was the compound or your peptide was eluted, and you are getting a particular peak. So now if I just show you, so as I have shown you in the method that we put in the product. So this is the product. So this is the precursor, sorry, this is the precursor and its product. So in the method, as I have shown, that you will be giving the precursor ions as well as all the product ions. So all these precursor and product ions are monitored, and we can see that how it is being eluted in a particular thing. So after that, we try to find out the CV and also the, so if the same range that we have seen in a discovery mode using fusion or QV, if it is like the up regulation or down regulation, so we try to monitor the same trend here, and that gives us what validation of a particular peptide or a better. Yeah, thank you.
All right, thank you very much, Ankit, Surbhi, and Amit. I think you have seen, various mass spec configuration. I just thought to update you that, more recently, last 2-3 years, proteomics has seen the paradigm shift of some very new configurations coming up, including the Orbitrap astral mass analyzer from Thermo Fisher, which utilizes 3 types of mass analyzers, like quadruple and Orbitrap and astral analyzer. And then now we have more recently coming from the Bruker, the timsTOF, which is another revolutionary technology, which looks into the a 4th dimension separation. Idea is in which way we can look at, in addition to the mass and charge and the intensity, can we also look at, and retention time, can we also look at the ion mobility as a 4-dimension separation? So therefore, this is one of the very revolutionary technology available, which utilizes the passive as a parallel accumulation serial fragmentation to look at how to select your precursors and increase your MSMS scan rate. So this is one of the technology which we are very pleased that we have the first setup of this in our startup company here, Proteomica International Private Limited. Dr. Arghya Banerjee, he's, I think, live available there. He's going to show you the timsTOF mass spec, and this is another mass spec configuration, which is, I thought, you have seen a couple of Orbitraps. Now you can see the timsTOF and appreciate, what is the latest revolution in this area happening. So, Arghya, please show the live session from your side. Start. So we can see you, but we cannot hear you. So
yeah, just a minute, Okay, so welcome everyone. So this is the India's first spin-off from the proteomics lab, like what you have seen, and we are standing at the Proteomica, and we have a very beautiful instrument which is timsTOF Pro at the Proteomica International Private Limited. So before moving, I will just give you a brief idea. These timsTOF Pro are the cutting-edge mass spec. Till date, the mass spec you are hearing are basically 3D. Either you work around on the intensities or the M by Z values or the RT value. Now, this is the first technology which works on the TIMS technology. Now, you just have to understand like how this works. Like you can consider 2 ships moving out. One has a bigger curtain. The one ship which has a bigger curtain can gather much amount of air, and it will be moved faster. Similarly, here in TIMS, we work with the cross-sectional areas. The ions which have the larger cross-sectional areas move out first, and the ions which have the smaller cross-sectional area leaves down. So there are 2 TIMS attached. One is the TIMS in and TIMS out. From one TIMS, the ions will move inter and get accumulated, and from the other TIMS, the ions will be moving out. So that is why it is called passive technology. which is parallel accumulation with serial fragmentation. So I hope I will not talk more. You have got the basic idea of what is the functionality of the things. So you can find, this is the cutting edge nano LC, which is the EvoShape nano LC. And you can find there is no sample collection vials. Okay. So what you have to do, there is a C18 material in these cartridges. These are the proprietary tips. You directly take this tip, put your sample inside, and directly place it there. And you can directly place it in the one of the matrix for this kind of activity. So once placed, you can directly open it. There is a syringe from where it takes the sample and it directly injects. It also has 3 samples, like 3 reservoirs. Most important thing is like it has 5 pumps. Instead of the local quaternary pumps, it has 5 pumps, and you can see like this is the high pressure pump, and all can be used for this kind of workflow. So very efficient, like you can run 400 samples per day. You can just put your samples on the tips and it will be coming out. From the sample collection mixture, it will be coming to the column oven toaster. What you can find, like this is my column, the 15 centimeter column, what you are seeing. It is kept in an oven test toaster so that the temperature is maintained at the 45 degrees Celsius. I will just close it, and this is my column. What you are seeing is the air filter or the vent from which the air is given out, and it does not require any special gaseous ion for that. So the air which is getting inside through the source, so this is called the nano captive source, and it moves inside. So the air which goes out, that behaves as the drag force for the ions. The larger the cross sectional area, larger it will move out. So here, like in this inside, there is a TIMS thing, and it is just a 10 centimeter long. And based on the TIMS separation, it is going to the top, like there will be a collision. Then this is the top, what you are seeing, it will be going to the top, and from the top it will be detected. You have to just find the robustness because processing saliva samples, serum samples are very challenging. With this kind of instruments, we can analyze with 5 minutes gradient with a 4 centimeter column. We are getting the counts of the proteins for saliva around 3,500. So you can see, so this is one another instrument which we want to show. This is the tissue bead box. So this is also one of the combinatorial technology which comes to. You direct have to place your samples into this append drops where this kind of magnetic beads are placed, and you can just process the samples. So these are all the cutting edge technologies what you can find. Like these are the magnetic beads, if you can see. So these all are the cutting edge technologies you can find, and we will be happy to help you and answer your queries in whatever ways you will ask. Please do ask your question. Back to you, Professor.
All right, thanks a lot, Dr. Arge, for a wonderful demonstration of our new setup, which I think will motivate all the participants to see a very balanced perspective of different mass spectrometer, which we are able to show them. So now broadly, I will continue. Now you have seen different type of nut and bolts of mass specs, right? Think about the quadruple, the Orbitrap, the ion trap, the TIMS, various type of analyzers. They can be put together for different applications, right? So one broad application is the discovery-based workflow when you do not know what you're looking for and you want to identify a new target. So therefore, you are looking at unbiased approach of quantitative analysis. There are 2 different principles involved. One is known as the DDA, data dependent acquisition. Other is DIA, data independent acquisition. And again, you are going to utilize these mass spec configuration for this. Additionally, once you know your target, you want to validate them, then you are going to utilize the targeted proteomics-based workflow using the triple quadruple or the Orbitrap-based configuration. So this is again giving you a variety of things can be done using these mass spec configuration, which we just showed you. So now you are able to generate some data from these mass spectrometer, how to identify the proteins, right? So that's the next thing what you want to learn. And more at the biochemical level, looking at more on the theory side, what we have these peptides, right? These are various amino acids come together with the peptide bond, CONH. And now with the collision energy, we are trying to fragment those. And therefore, with the amino terminal and the carboxy terminal side, these ions are going to get fragmented at the peptide bond. And as a result, they're going to generate some B and Y ions. This B and Y ion, so the ion which is retained toward the amino terminal is known as the B ions. More closer to the carboxy terminal will be Y ion. But for each pair, you will understand there will be both B and Y ions. So the closer to the carboxy will be Y1, B4, and the closer to the amino terminal, B1 and Y4, right? So likewise, series will be generated. So how to analyze this data and look at these concepts which I just talked to you, one could go to this particular software interface, matrixscience.com, and this is one of the free open access software available, where you can utilize at least the basic features for doing the protein identification. So therefore, you can now think about how to utilize this software for data analysis for protein identification. So I want to now invite Surbi from our team to give you the live demo of the mass spec data analysis for protein identification using mascot. So over to you, Surbi.
Yeah, thank you, sir. I'm sharing my screen. I hope my screen is visible. So I'm going to give a demo of a simple web-based software that is the Matrix, that is on Matrix Science website. It is Mascot. And for very simple and small MS files, for not very complex samples, maybe just 2-3 protein, a mixture of 2-3 proteins or something
so just
like
to add
that.
here, Surbi, before you start, we have also shared a link in the Zoom to the participants if they want to download a file and want to, do the analysis on their own. While Surbi is giving the step, you can also do that, or you can learn this and you can analyze it later on. But we have shared a file which you can access that. Please continue.
Right. So the file that has been shared with you, there is one .mgf file which can be used with this particular software. For the analysis, you'll have to go to matrixscience.com, and here you can access Mascot server. Now, there are multiple things that can be done, but what we are going to do is the MSMS ion search. So go to perform search. Now, you'll have to put in your name, email ID, search title. If you're using it for the first time, you will be redirected to verify your email ID. So the first time you do, the results won't come up. So then you'll have to verify your email ID and then do it again. Since I've done it multiple times, it's going to happen in the first time for me. Now, the most important thing for a proteomics data search is the database. Now, you need to tell the software against which database you want to search your raw file. So, this could be the source organism of your sample, or maybe a combination of organisms, depending on your experiment or depending on your sample. Contaminants database is one thing that we generally use because we don't want to identify any peptides or any proteins that can come as a contaminant. Now, while working in a lab, there are several proteins that can come up in the samples, like keratin or BSA or anything like that. So we don't want to identify that. So for that, we use a contaminants database. For this experiment, we'll use the SwissProt database. It is a database of all the proteins that we know of, the sequence of all the proteins. But in case of any specific experiment, we can use any other specific database also. Next is taxonomy. You can mention. Now, I'm going to skip it for now. Enzyme, select the enzyme with which the sample was processed prior to the generation of the data. Most commonly, we use trypsin. So I'm going to use trypsin. Now, miscleavage. Any enzyme is not 100% efficient. There will be some sites where the cleavage will be missed by the enzyme, trypsin in this case. So how many missed cleavages do we want to allow? 0 is very stringent, and as we go up, the stringency is reduced. So we'll for now mark as 2. We can allow up to 2 miscleavages. Then there's an option to quantitate also. If you have used any labeling technique for quantitation, but right now we are not going to use. Then again, for cross-linking also, there are options if you have done cross-linking in your samples. Now, then comes modifications. There are 2 types of modifications, fixed and variable. Due to sample processing, some modifications are introduced into your samples, and which this we need to take into account. So fixed modification is the carbamidomethylation of the cysteine because of the reduction in alkylation of the cysteine. So we are going to add this. And another modification, which is a variable one, is the oxidation of methionine. In addition to this, if you are looking at any other post-translational modifications like phospho or anything else, you can select that also. For now, we'll just select oxidation of methionine. Next, the peptide tolerance and MSMS tolerance, these are instrument specific. We can keep it as default for now. Then we select the data file over here. As I said, the file that has been shared with you, the .mgf file, is compatible with Mascot, and you can use this for your own test. And if there is any other file, you can see what all files are compatible with Mascot. So Mascot generic file, which is the MGF file, NZML, micromass, instrument, whichever instrument was used to generate this data. For now, we keep it default, and target FDR, which is the false discovery rate. It's best to keep it 1%. If you want to go more stringent, you can go lower. Then we start the search. So the Mascot is pretty fast. You will get the results in a matter of a minute or 2. And this is how the result page looks like. Here you will see all the details that you have put in, your parameters. And here we get the results. So you can see that 4 protein families have been identified. And these are the protein families identified. So you can see that, let's say the first protein, TIC40, this is from the organism Arabidopsis thaliana. If you click on this, then you will see the peptide that has been identified for this particular protein. Let me click on this one to generate a report, a protein report. Yes. So here you see this particular peptide mapped onto the entire protein sequence. So you'll get the location of the peptide and the coverage. So 6%, this peptide is covering 6% of this protein. Yeah, so another important thing is this score. This is the Mascot score, and it tells about the possibility of whether this peptide is an actual match, or is it just a random match. So higher the score, higher is the probability that the peptide match is an actual match and not just a random match. So you can explore the results and see what all is there. Another thing that I want to show is, if you go to this peptide, here you will see this number written 51. If you click on this, this will generate a peptide repeat. And here you will see that in this particular file, and for this particular peptide, what all B and Y ions, as Sir just mentioned what B and Y ions are, what all ions are identified for this particular peptide. Here you will see the table of the MYZ ratios of those particular ions. And yeah, so this is how you can analyze a simple sample using Mascot. You can explore around and see what else are there, but this is a brief summary of it. Thank you so much.
I think it was very clear, in a very short time. Please unshare your screen. I can now continue. So, all right. So I think what participants would have seen by now, that the configuration of these mass specs and how one could operate them and how one could analyze their data just for protein identification. But now let's move on to the protein quantification part. So there are different ways. Now, since you know the mass spec configuration, it's much faster for us to now move forward. Now you are going to look at different ways of quantification. So idea is, can we look at either labeling the protein in some method, or without labeling the protein and still want to do quantification. So there are in vitro approaches where one would look for the amino acid based labeling in the technique known as the isotope coded affinity tagging, or one could look at the peptide itself to be labeled in the N-terminal labeling with the methods known as the isobaric tagging for relative and absolute quantification I-track or tandem mass tag TMT, or look at the C-terminal labeling with the proteolysis 16O-18O pair, or one could do in vivo labeling with the SILAC. So these are various methods which I told you about, the label-based method. When you are looking at some way of label, metabolic or chemical labels, and want to utilize those labels to look at the quantification that are on in the mass spec. Or there's a new concept when one is look for even the label-free method. It means you're not labeling, but you are looking at the peptide quantification and their intensity or spectral count. So I'll spend a few minutes time here on these methods. So let's say I-track, isobaric tagging for relative and absolute quantification, is a very popular method, or similar technique also known as the TMT, tandem mass tag. Here you're looking at some tag. Let's say these are the 4 conditions you want to compare. Your 0 time point and time point 1, 2, 3 in more like temporal expression you want to look at, for a biological sample. You have added in 4 conditions. After taking out a sample, you extracted the protein, digested to make peptide. These peptides you are labeling with 4 types of tagging. So 114, 115, 116, 117 are the reporter ions. And then the balancer region is 31, 30, 29, 28 to make it all 145, which is isobaric. So it means you don't want to have anything different mass on all the 4 conditions. So therefore, the I-track is the isobaric tagging methods. After labeling all of them, you have mixed them together, then run in the mass spec, and after MSMS fragmentation, then you are able to see the reporter ions. So theoretically, what it looks like, you have taken 4 conditions. After digestion, you got peptides, labeled them with I-track labels, analyze them in the mass spec. After MSMS, you see signature regions of I-track 114, 15, 16, 17. This can be utilized for your peptide quantification and for the MSMS for the protein identification. So this is where one could look at directly with the mass spec, how to do the quantitative proteomics. Additionally, as I said, one could also look into, without labeling, can I just look at the how much peptide abundance is there, their spectral count is there, or the intensity change is there. The concept is, if like in the chromatograph is reproducible at every given retention time, whether it's a control condition or the treatment condition, same composition of peptide will be eluted out. So therefore, you're running every sample in a very stringent same condition, but individually, one after the other. You are not pulling anything here. And therefore, after each run, you are generating the entire chromatogram of that particular sample. And then you are going to compare what is the peak intensity of the lower or the higher in control and the treatment condition. So this method is known as the label-free quantification. But both of them come under the DDA, or data-dependent acquisition, which looks at the most abundant top precursors and try to look at those precursors and their further fragmentations. For example, the blue, red, green ones, yellow ones, these ones are the most intense precursor which are being fragmented, and one is generating the MSMS from that. Only problem in this case is lot of missing value which comes as a part of the data. So therefore, now the new field is emerging, which is known as DIA, data independent acquisition. Here we are looking at survey scan across all the isolation windows of 300 to 1300 M by Z, and you are trying to get a snapshot of each precursors of different M by Z region. So you are not having that much missing value problem. This is another newer approach of looking at the quantification. So now, since we theoretically got at least some idea about both label-based methods, includes the I-track, TMT, ICAT, SILAC, many methods are there, or label-free methods. All of them are together as a DDA, and a new approach known as DIA. So based on that, you are able to now do a lot of quantitative proteomics and compare your control versus disease condition at the proteome-wide scale level. So now in just next few minutes time, Amit will at least give you a flavor of a software, MS Fragger, which all the participants here they can also use in their own laptop, which is the open access software. And timing is short. Amit will just show you the interface of it, how the software looks like. And we have some other session where you can follow up on the data analysis part. So Amit, please show the MS Fragger software now. Please unmute and speak. Amit, you have to unmute. All right, please continue. Timing is very short.
Yeah. So coming to the one of the softwares that we use for as in the mascot you have seen key we use mascot for the primary from the simple mixture of protein samples. So for mass spec generated complex files we use softwares like FracPipe. So whatever the raw data is generated, we use FracPipe softwares to analyze the data to get the what are the different proteins are identified from the raw files. Right. So here's the interface of the FracPipe. Coming to the workflow. So this is basically the entire interface of the workflow. For any kind of DDA approaches, we basically try to set a workflow of level 3 quantification. Here is if we want to add the raw files here we need to add the raw files. So these are the different raw files that I am going to analyze. You just need to select them. Now you can also give the experiments and the bioreplicates too. I am keeping it by the parent name and the bioreplicates. Now coming to the most important thing is the databases. So here we are using a human database. So for that you need to download the database, or if you have already a database there, you can also upload the database here, and you need to add decoy. So decoy is basically a non-existent protein sequence database that will help you to understand what are the falsely discovered proteins are there. So basically to eliminate the false discovered protein, you need to add the decoy database. Now another important thing is the MS Fragger. I think there are some technical issues. I am not able to switch the screen here.
Okay, so timing is short, Amit. What we can say that the students who are interested in learning more about this particular software, we have some other sessions which we can share with them. But this is one of the very user-friendly software which can be taken forward for quantitative analysis. Amit, please unshare your screen. And then after this, one could try to utilize other software like MetaboAnalyst or the Perseus, and try to use them for the quantitative analysis. Amit, please unshare your screen so that I can share my screen now.
Yes.
Okay, so just to summarize what we learned today, essentially idea was to make all of you familiar with mass spectrometry based workflow, in which way one could use different type of mass spec configuration to look at protein identification and protein quantification. I think you must have been, felt excited that you saw several cutting edge mass spectrometers, which can do various type of analysis from the discovery to the targeted analysis. And broadly, these are the concepts which you should keep with you. The MS-based quantitative proteomics now involves both DDA and DIA approaches. DDA involves label-based quantification or label-free quantification. Label-based idea is you are adding some tag which you are going to utilize as a label for looking at MS level, whether it's tandem mass tag or the I-track based reporter ions. Label free, we are looking at only the abundance of these peptides and their spectral count. All of them are looking at the most intense precursors only. So they are missing out the less abundant precursor. Therefore, the newer approach like DIA has come forward, which looks at the entire M by Z window to do the entire scanning. And then therefore, we are overcoming many of the missing value problem which we see in the DDA-based data. So DIA is currently the major workhorse for quantitative proteomics. The different software which are already available, both commercial and open access. But open access software like MS Fragger, which I just try to give you a glimpse of it, as there are the new software for the DIA analysis known as the DIANN software. These are currently the buzzword in the field. You should definitely try to explore these softwares, and you can. The best part here is it does not require the extensive RAM or the very high configuration. You can do on your own laptops, and at least get a flavor of it. We'll be happy to share you some data files for your further analysis and your further, if you want to try on your own. But that's all from our side to give you how mass spectrometry-based quantitative proteomics you can utilize for addressing many problems. And as you can say, to reach out to the final biological conclusion, there are many technical routes which you have to follow. Depending on what your project design is, what your sample size is, you can choose one of these paths. In the process, once you know the mass spec basic workflow, once you know the nut and bolts, once you know the software and the workflows for these analysis, I think you'll be better to get the good outcome from your biological problem which you want to understand, and you will have much more concrete idea for the protein which you can take forward for your next set of hypotheses for the validation as well as further understanding and characterization. I think that's all from our side today. Let me thank all the TAs here, Surbhi, Ankit, and Amit for showing you a few interesting instruments and the software. Of course, a lot can be learned in this area. So you're encouraged to interact with us again in future, and we'll be happy to guide you towards your proteomics journey. Thanks a lot, everybody. And if you have any more query, we'll be happy to take it forward. But right now, I think we are overshooting the time. So I'll stop the session here, and we'll close this thing for the today. Okay. Thanks a lot. Bye, everybody.